Hello everyone and welcome to Roadmap. So we are continuing with the topic reproduction in plants part 3, and if you like the video, don't forget to click on thumbs up button below. You can also subscribe for more videos and updates, and you can also share, and you can also give your suggestions in the comment section below. The topics to be covered are introduction, reproduction in plants, asexual reproduction. In that, we'll learn about binary fission, spore formation, fragmentation, vegetative propagation, sexual reproduction. And in sexual reproduction, we'll learn about the flowering plants, how the pollination takes place, the types of pollination, and then the fertilization and fruit and seed formation. So coming to the introduction, I'll just give a glimpse of it that what we have learned. We have learned that life processes and are involved and how the systems are very important and essential for organisms to survive and grow. So basically, we have learned so far about the different systems, and so the reproductive system is also one of the system which is very essential, which gives rise to offspring, and then it is essential because it cannot give a specific species will give rise to a specific species only, not to some other species. Like chimpanzees won't give rise to gorillas, and gorillas won't give rise to chimpanzees. So basically, there are some sexual methods of reproduction that takes place, which we have already learned about budding, which takes place in yeast, and then we learned about the fragmentation and spore formation, and then we learned about the binary fission, and we learned about the different types of sexual reproduction that takes place, and then we have also learned about the vegetative propagation, what it is exactly, like the sexual reproduction which takes place in presence of leaf, stem, or root. It is nothing but a vegetative propagation, and there are some natural methods of vegetative propagation that takes place, like vegetative propagation by leaves and then by roots. It also takes place by rhizome, bulb, CORM, and then it also takes place by tuber. So we have already learned about this so far, and I would suggest you to go back and check the video of that previous video, whatever we have learned, and it will give a better understanding for you, and your conceptual clarity will be better for that. So coming to the next topic now, that the artificial methods of vegetative propagation. Artificial methods of vegetative propagation is nothing but the process of growing new plants using man-made methods, and it is simple, faster, and less expensive method for multiplying plants. It is for this reason that this method is commonly used by agriculturalists and horticulturalists. Some artificial methods of vegetative propagation are cutting, grafting, layering, and tissue culture. So we'll learn about them one by one in more detail. Now coming to the cutting, in this method, a healthy young branch of a plant having leaf buds is cut off and planted in moist soil. In due course, the branch develops roots and grows into a new plant. Sugarcane, rose, and grapes are grown from cuttings. So the examples of cuttings are nothing but sugarcane, rose, and grapes. Coming to the grafting, it is the commonly practiced method. A new variety is obtained from the mother plant, and in this process, a detached part of one plant is inserted into the stem or the root system of another plant. So the short piece of detached shoot containing several buds is called scion, and the lower portion of the plant that is fixed to the soil by its root system is called the stalk, and the scion is cut and placed face to face with stalk. The scion and stalk are then firmly tied together. They establish vascular connection with each other after a few days, and this technique is very helpful in producing desired varieties of plants and fruits. And it is common plants which are grown are mango, guava, apple, and pear. So just what you have to remember in grafting is that the scion of one is cut and it is grafted with the another stock, so that you will get the variety of plants in this way. So this is all about the grafting method. Now coming to the layering, in this type of method, a young branch is bent towards the ground and covered with moist soil. After some days, new roots develop from the covered part, which are in contact with the soil, and this is called as layer, and this process is called as layering. The branch is then separated from the parent plant and allowed to grow into a new independent plant, and cherry, jasmine, grapevine are propagated by this method of layering. Coming to the tissue culture, Basically, from the diagram, I'll explain you that a tissue sample is taken from a parent plant, and this is called as explant. Then, this tissue is placed in the agar growth medium containing nutrients and auxins. Basically, the agar medium is prepared with the nutrients like copper sulfate, and there are some other nutrients. There are some micronutrients and macronutrients, and these are taken with the fixed proportions, and then the auxin cytokinin with ratio is maintained, and they are allowed to grow in the medium. And further, the samples are developed into, then they are with the process of aseptic conditions, like LAF is used, laminar airflow is used in this process, and where the with the help of aseptic conditions, so that the contamination should not be taken place and the explant should have a healthy growth. So they are in that, with the help in LAF condition, with the aseptic condition, or between the two burners it is done, and it is transplant from the explant is processed into the nutrient medium, and then further the samples develop into the tiny plates, and it starts growing. And then further what happens, that the explant, it also has the other procedures which are done, like the explant is taken, and then if one explant is taken, it will grow into nutrient medium, and again they are transferred to other bottles or another plates, and then again they give rise to more plants. So in this way, the one plantlet give rise to many plants, and the plantlets planted, they are composed, they are into compost, because this is generally the greenhouse, poly house we can call it, where the conditions are maintained, that it is the dark room, so that the plants will able to grow under certain country conditions. And then then exactly many plants are developed from one explant. So this is what happens exactly in tissue culture. It is a method. A piece of tissue is cut from the plant, and then it is kept in nutrient medium under control condition, and the tissue grows into a mass of cells which is called as callus. Small pieces of callus are put in a different nutrient medium, which makes them develop into new plantlets. These plantlets are then transferred into moist soil for further growth, and plants like orchids and asparagus are grown by this methods. There are also other fruits are also grown, like banana is grown in this condition. Basically, this method is very cheap to carry out, and you will be able to get many plants from one explant, and it is a very fast and simple process. And in this process, what happens exactly, even if the seasonal fruits we are not able to get it naturally, still by some artificial methods we are able to grow with this seasonal fruits and vegetables. So this is how the tissue culture is done. Now there are some tissue culture, these are some advantages of tissue culture. Now coming to the advantages of vegetative propagation, the daughter plants are identical to the parent plant, and the method is quick, economical, and dependable. The tissue culture can be carried out throughout the year irrespective of the season, and they are useful for growing plants which do not produce seeds. And now coming to the disadvantages of vegetative propagation, it does not produce any variation, and therefore new varieties are not produced. Possibility of dispersal of plants is very low. So this is all what happens with the vegetative propagation, as they are man-made methods. So they are easy and they are available throughout the year, and it is not that time consuming in comparison. So this is all about the asexual reproduction methods, and natural methods and artificial methods. And I hope that you are liking the video, and stay tuned for more lessons to come. Thank you for watching.